Hello everyone, welcome to my channel Bioscience Lessons. Today we are going to talk about glucose transporter. So when we take carbohydrate rich food, it is digested and it is converted into glucose in the stomach and the small intestine. After that, glucose is absorbed through the small intestine into the blood, and from blood it will be transported across the body. And this glucose is very important because it is used for glycolysis process and for the ATP production. So glucose is very important for body. But problem is glucose is a hydrophilic molecule. That means it is a polar molecule. It cannot easily cross lipid bilayer of the cell membrane. So it has to be transported with the help of some transporter, which is called as glucose transporter. Glucose can be transported by two carrier, GLUT and SGLT. GLUT works by the process of facilitated diffusion and SGLT works by the process of secondary active transport, which is present mostly in intestine and kidney. As I told, there are two types of glucose transporter, GLUT and SGLT. This GLUT is sodium and ATP independent, means it does not require sodium, it does not require ATP hydrolysis. No energy is used for this process and it is a passive process. It is unipore, that means only glucose is transported. No any other ions or molecule will be transported, only glucose is transported. Next, it is done by the process of facilitated diffusion. What it means is, suppose this is a cell membrane and this is the carrier molecule GLUT, then glucose will be transported from the region of high concentration to the low concentration. That's why it's called facilitated diffusion. Facilitated means this carrier will be facilitating the transport of glucose, diffusion of glucose from the region of high concentration to low concentration. Next, there are 14 different types of GLUT which is present in the cell membrane. All are integral membrane protein and they consist of 12 transmembrane domain with both N and C terminal towards the cytoplasm of the cell. Suppose this is the cell membrane, this is the outside of the cell and this is the inside, then this GLUT will pass the cell membrane 12 times and both N and C terminal will be present inside the cell. Next we will talk about SGLT. SGLT is sodium dependent glucose transporter. That means sodium will be transported along with the glucose. That's why it's called co-transporter and symporter. Symporter means both glucose and sodium will be transported in the same direction and it is done by secondary active transport. Suppose this is the cell membrane and this is SGLT, then sodium will be transported from region of high concentration to the region of low concentration and along with this glucose will also be transported. And both are transported in the same direction. That's why it is called as symporter. There are 7 different types of SGLT which has been identified, but 2 are very important. First is SGLT1, which is present in intestine near the lumen side, and second is SGLT2, that is present in kidney in situ of the nephron. SGLT is also an integral membrane protein and it consists of 14 transmembrane domain, that means 14 times it will pass the membrane. Next we will talk about various types of GLUT and SGLT. First we will talk about SGLT1. SGLT1 is present in intestine towards the lumen side and it is involved in glucose and galactose absorption. We will see the diagram. This is a diagram of SGLT1. It is present in intestine towards the lumen side. This is the lumen of the intestine and these are the epithelial cells of intestine which is called enterocytes. This is the basal part of the intestine and here interstitial space is present and here blood capillaries are present. This SGLT1 is present in the lumen of the intestine. It transports sodium from the region of high concentration to the region of low concentration. Along with this, glucose will be absorbed. This glucose will be used for ATP production by the process of glycolysis and excess of glucose will be transported towards the basal part and it will move to the blood. Here, GLUT2 carrier molecule is present which is involved in transport of glucose out of the enterocytes. This excess of sodium will be transported outside and to balance this, potassium will come inside the cell by the help of sodium potassium pump, and it requires ATP hydrolysis. This SGLT1 along with glucose, it also transports galactose. Talk about SGLT2. SGLT2 is present in kidney in the proximal tubule of the nephron, and it is also involved in glucose reabsorption. This is the diagram of SGLT2. It is present in kidney in the nephron. This is the proximal convoluting tubule of nephron. This is the lumen side and these are the renal tubule epithelial cells, and these are glomerulus. Here blood will come and it will undergo filtration. In this lumen, filtrate will be formed which will contain water, glucose, sodium and various other ions and waste products. This SGLT2 is present in the lumen of the PCT. It will transport sodium from the region of high concentration to the region of low concentration, and along with this glucose will be transported inside the cell, and this glucose will be then transported into the blood with the help of GLUT2, and this sodium will be transported into the blood, and in exchange of this potassium will come inside, and it requires sodium potassium pump which requires ATP hydrolysis. So here active process is happening, and in SGLT2 secondary active process occurs. Next we will talk about various types of GLUTs. First, GLUT1. GLUT1 is present in blood-brain barrier, brain, RBC membrane and various other important organs. It is important for the basal glucose uptake, and it has low Km value. Low Km value means high substrate affinity. That means GLUT1 will have high affinity for glucose. So when glucose is present in low concentration also, this GLUT1 will be involved in transport of glucose. That's why it is used for the basal glucose uptake. Next is GLUT2. GLUT2 is present in beta cells of pancreas, and these beta cells of pancreas are involved in insulin secretion. Next, which is also present in liver, kidney and in the intestine towards the basal side. We have seen in SGLT1 and SGLT2 diagram that GLUT2 will transport glucose out of intestinal epithelial towards the blood. This GLUT2 has high Km value, that means it has low affinity for glucose, which means when glucose is present in high concentration, that time only GLUT2 will be involved in transport of glucose. Because of this reason, this GLUT2 acts as a glucose sensor in the beta cells of pancreas and help in insulin secretion. And it is also involved in transport of glucose out of intestine and renal epithelial cells. Please see the diagram. So this is GLUT2 which acts as a glucose sensor in the beta cells of pancreas. This is the beta cells of pancreas. When glucose is present in high concentration in blood, this GLUT2 will transport glucose into the beta cells of pancreas, and glucose will undergo glycolysis. And due to this, ATP will be produced. When excess of ATP is produced, it will block the K plus channel, that is potassium channel, and it will cause cell depolarization. That means cell will become less negative, and due to this, potassium channel will open and calcium concentration will increase in the cell, and this will cause the vesicles which contain insulin to undergo exocytosis, and insulin will be secreted in the blood, and this will be transported to adipose tissue and skeletal muscles, cardiac muscle, and this will translocate GLUT4 on these cells. We'll see later this GLUT4 will be present inside the cells, especially adipose tissue, skeletal and cardiac muscles, and when this insulin bind to the insulin receptor in these cells, this GLUT4 will be translocated to the membrane, and this will be involved in glucose transport. So glucose will be transported from the blood inside the cells, and due to this, blood glucose level will decrease. We'll see it later. Next, this GLUT2, it is present in the basal part of the enterocytes. This is enterocyte, that is intestinal epithelial cells, and this GLUT2 will transport glucose, galactose as well as fructose from the enterocyte into the blood capillaries. GLUT3, it is present in brain, placenta, kidney, and it is similar to GLUT1. It is involved in basal glucose uptake and it has low Km value. Next is GLUT4. GLUT4 is present in skeletal muscles, cardiac muscles, adipose tissues, and it is involved in insulin dependent glucose uptake. Only GLUT4 is insulin dependent, rest all GLUT and SGLT are insulin independent. One more thing, all the other SGLT and GLUT are constitutively expressed in the cell membrane, but only GLUT4 is present inside the cell in the vesicle, and it comes to the membrane only when insulin binds to the insulin receptor which is present in the cell membrane of these cells which contain GLUT4 vesicles. Suppose this is an skeletal muscle cell and these are vesicles which contains GLUT4. Currently it is present inside the cell. It will come to the membrane when insulin binds to the insulin receptor. So this is the insulin receptor which is present in cell membrane. This consists of two subunit, alpha and beta subunit. Alpha subunit binds to insulin and it undergoes dimerization, and because of this, beta subunit which contain tyrosine residue will undergo phosphorylation and gets activated. This insulin receptor is also called as receptor tyrosine kinase. So it has tyrosine kinase activity. It will activate phosphatidylinositol-3 kinase and it will cause this vesicle to undergo membrane fusion, and because of this, GLUT4 will be translocated into the cell membrane, and this will transport glucose from the outside of the cell to the inside. When this insulin is removed later and signaling stops, this will again undergo internalization. As I told, when blood glucose level goes very high, that time beta cells of pancreas have GLUT2 which is present in the membrane, and it will transport blood glucose inside the beta cells of pancreas, and it will cause insulin secretion, and this insulin will then come and bind to the insulin receptor which is present in the membranes of adipose tissue or skeletal muscle or cardiac muscle, and this will cause the GLUT4 to be transported to the membrane, and this GLUT4 will then transport glucose from the blood inside the cells and decrease the blood glucose level. One is GLUT5. GLUT5 is present in intestine towards the lumen side and it is involved in fructose transport. So as you can see, this is the enterocyte, that is intestinal epithelial cells, and this is the lumen side of the intestine, and this GLUT5 is present towards the lumen side of the intestine. It is involved in fructose transport, and this GLUT2 will then transport glucose, galactose as well as fructose towards the capillary. So that's it for the video. Hope you like this video. Please subscribe to my channel. Thank you.